Assalamualaikum. We are from group 11 and today we are going to do a recombinant DNA by combining a targeted gene from cell DNA into a plasmid, therefore creating a recombinant DNA plasmid. So first of all, we need to have cell DNA which contain the targeted gene that we want to have into our plasmid. So we have this long tape, yellow paper, which is the cell DNA, and this is the targeted gene that we want to insert into the plasmid, in which it is protein insulin gene. And after that, we want to have the which is the host. So next, we want to have the recipient, which is the plasmid DNA from bacteria. And this is the plasmid that we have. We already set up for it to be in a circular shape, as in plasmid DNA. And in this plasmid, we have the origin of replication gene site. We also have the 3 antibiotic resistant gene that we have here, which is kanamycin, tetracycline, and also ampicillin resistant gene on this plasmid. And we also have need restriction enzymes as the key for it to cut the DNA plasmid and also the cell DNA, in which the mechanism used is the cutting and pasting. So we need to have the scissors. So the scissors is actually the restriction enzyme for it. So we have many types of enzyme here, and from all of this we can only choose one enzyme that can cut 2 restriction sites on the cell DNA and 1 restriction site only on the plasmid DNA. So we can try to choose either one of them from all of this.
So out of 8 enzymes we have here, we can only use 2 that is matching to our cell DNA and our plasmid DNA. Say, okay, the 2 matching are the BAM-H1 and also HPA2. Okay, amongst the 2, we choose the HPA because the HPA cuts the cell DNA in a closer distance to the targeted gene. So we will reject the BAM and we take the HPA enzyme. Okay, here we are at the cell DNA, and this is our restriction site, the first one. Okay, so here comes the enzyme, our HPA2 enzyme, and thus, okay, now the enzyme will cut our gene apart. Okay, so this is the unneeded DNA. Okay, so this is our second restriction site on the targeted cell. Alright, so here comes our enzyme. Okay, and here the enzyme will cut apart this DNA, and thus we will have our targeted gene. Okay, now we're moving on to our plasmid DNA. Okay, so the site of restriction site is here, and our enzyme will come right in and cut apart our plasmid DNA to open the site. Okay, now that our plasmid DNA is cut, okay, this is our targeted gene and this is plasmid gene. Therefore, it will our ligase enzyme will come to connect our both genes. So when because they are matching, so they can join together, and then the ligase enzyme comes and connects our genes together. Okay, now this is the same for our second site, where the ligase will also connect the gene. So when both are connected, we will have a circular gene again.
The recombinant DNA from the cell DNA, the targeted insulin gene, and recombine it with the plasmid DNA that we have that have all 3 antibiotic resistant genes. So we have the recombinant DNA to test whether the recombinant DNA is done. We need to put it in a medium where an antibiotic can be used to indicate whether the recombinant DNA is done or not. So since in the plasmid we have all 3 restriction enzymes, all 3 antibiotic resistant genes, we can use all the 3 antibiotic resistant genes, tetracycline, ampicillin, and kanamycin on the medium. And if the plasmid DNA are detected and not die, that means that the recombinant DNA is done. And that's all from us. Thank you.